one moment. Excellent. Thank you very much, Diane, and thank you to the organizers for inviting me to speak with you today. I actually wanted to speak about ion channel targeted therapy for a rare kidney disease, and so that's what I will be discussing today. These are my disclosures relevant to my talk today. Is my financial interest as an founding advisor to Goldfinch Bio, a company which is just around the corner from here in Kendall Square. So I wanted to start by just framing the problem of why anyone would be interested, A, in ion channels, and B, in kidney disease. And it's really to frame the problem to say that about 1 in 9 Americans have chronic kidney disease. It's about 800 million people worldwide, and it's actually a problem that continues to rise. It is also a very expensive problem. In this country, we spend about $110 billion per year to take care of patients with kidney failure. That is more than twice the entire NIH budget for all research in all areas and all diseases. It's really a mind-boggling number, and there's a very dire need for therapeutics. And yet, unfortunately, this field is dead last in therapeutic development across all fields of medicine. So there really is a very significant need for treatments in this space. Unfortunately, for various reasons, which I will not have a chance to discuss today, therapeutic development in this space has lagged. A lot of it has to do with the complexity of treating kidney disease as a singular entity rather than a collection of discrete molecular mechanisms. And so, for example, in contrast to cancer or immunology, the kidney space is just catching up to that idea. One of the ways that one could imagine building therapeutics in an area where there aren't any would be to start with something that's a little bit easier to understand. So the premise that I started with is the idea that perhaps attacking the problem from an orphan, rare, monogenic disease might actually provide an easier target, an easier understanding of what the relevant pathways might be. And then we've known many examples where even that focus on a rare disease can ultimately have transformative potential for other diseases as well. And I just wanted to provide a quick example. Mike Brown and Joe Goldstein, of course, famously have also received the Nobel Prize. But they didn't actually focus on treating high cholesterol, which is perhaps arguably one of the most successful medications built to date. But actually, they wanted to treat a 6-year-old girl who had rolled into the NIH suffering from a heart attack. And in trying to understand the underpinnings of her disease, they discovered the LDL receptor, and the rest is history, you could say. So this just speaks to the fact that potentially focusing on rare disease can be beneficial. And so that's one of the departure points for the story that I wanted to tell today. The disease that I wanted to speak about is called FSGS. It stands for focal and segmental glomerulosclerosis. It's a rare kidney disease that has no cure. It affects children as well as young adults. And it's rare, as in the United States, the we have is that it's 1 in 2,000 people. So it's a devastating disease that leads to kidney failure. It is primarily caused by a lesion in a particular cell type in the kidney, which is called the podocyte. Podocytes were very interesting to me because I came from hippocampal neurons into podocytes. I'm about to tell you the story. Because they're the only postmitotic cell in the kidney. All other cells can regenerate, but there's no known stem cell or other way to replace one's podocytes. We're all born with a certain number. And when the podocytes die, the kidney filter essentially dies or collapses, and that leads to kidney failure. One way to demonstrate where the podocytes lie in the kidney filters, just to orient you, if this is, okay, we'll go with this. If this is a kidney, and you take a small snip of a kidney, this is the glomerular filter unit of the kidney. Blood comes in, gets filtered through this structure, and the cleaner blood comes out. If you zoom into this area, here you can see that around this glomerular capillary, there is a basement membrane upon which sit these podocytes with the interdigitating foot processes and structures in between those interdigitating foot processes that form a basket weaving structure ingeniously built by nature to form a filter. So when the podocytes die, as demonstrated here, that is essentially the point of no return. So I'm saying all of this to demonstrate that the logical therapeutic hypothesis would be to develop a therapy that would be podocyte preserving. At the present time, there are no such therapies, and so that's where the story begins. Now, this is where the ion channels come in. The reason that I went from hippocampal neurons into podocytes is because in 2005, Michelle Wynn and her colleagues at Duke published a channelopathy, mutations in TRPC6, gain-of-function mutations in TRPC6 that lead to FSGS. That was the reason that I got into the field, but that turned out not to be the channel that I focused on. So in trying to understand all of podocytes and monogenic causes of podocytopathies, I discovered that while TRPC6 mutations are definitely there, and the plot will thicken in a moment about TRPC6, there were several other more common, within the context of a rare disease, more common mutations that coalesced, several autosomal dominant as well as recessive mutations that coalesced on the activation of RAC1. Now, this was particularly interesting to me for a couple of reasons. Oh, I should mention that the demonstration that these mutations are relevant to humans, or that the activation of RAC1 is relevant to the disease, is not only from the human genetics, but also from the fact that hypertensive proteinuric rats also have a notable activation of RAC1, which suggests that the genetics and the biology are on the right track. There was a slew of evidence to suggest this, as also well reviewed here, that there were several upstream signals that coalesce on the activation of RAC1 to cause ultimately proteinuria, which is the faulty kidney filter, the spilling of essential proteins in the urine, and the development of FSGS and kidney failure. Okay, so the reason I was very fascinated by the activating RAC1 mutations is because the two places where we had previously done some work in relation to RAC1, one of them was in the brain, was in relation not to TRPC6, which was actually the negative control for this experiment, but actually TRPC5. So TRPC5 shown before, the mic goes in and out a little. Raise it. This? Oh, use this one. Oh, you have. Okay, good to know. Can you now switch to that? Okay. So we had done some previous work to demonstrate that the activation of RAC1 is responsible for the vesicular insertion of TRPC5 ion channels in the plasma membrane. TRPC5 is most highly expressed in the brain, where I thought it would have a role, but also secondarily in the kidney, which I had relatively ignored at the time that I was doing this work. But it turned out that actually podocytes express not only TRPC6, but also TRPC5, which is of relevance. And so, you know, there we were kind of intrigued by the fact that activating RAC1 mutations don't seem to cause any other trouble except for kidney disease. And here we are with a potential link to TRPC5 as a putative target for this pathway. One could, of course, ask, why not target RAC1 if you're trying to treat this disease? And the answer would be, every time we have a cut on our skin, we rely on RAC1 in order for the fibroblasts to crawl and close that wound. So you probably wouldn't be prudent to do that. There are probably calcium mechanisms that are controlling that process as well, not always dependent on TRPC5, but TRPC5 appears to have a role in relation to RAC1, at least in the brain as well as in the kidney, which was of relevance to this story. So now I'm going to kind of pole vault through a lot of work that we did in this area, but just to demonstrate that we basically focused on TRPC5 and TRPC6 in podocytes and demonstrated that indeed it is the activation of RAC1 that is responsible for the vesicular insertion of TRPC5 in the plasma membrane of podocytes, not TRPC6. But intriguingly, there's a positive feedback loop that is set up. So the calcium that comes in through TRPC5 channels is actually responsible for the activation of RAC1 in this system. And so what ultimately happens when you have the activation of TRPC5 and RAC1 disease circuit, as I call it, or disease pathway in podocytes, then you end up with this phenomenon, which is completely detrimental to podocytes. It's a collapse of their cytoskeletal integrity, which ultimately would lead to the podocytes falling off the filter and the eventual development of disease. This was work that we did in vitro to demonstrate this. We then used a specific, actually non-specific, a TRPC4/5 inhibitor that came out of the MLPCN program called ML204. And as you can appreciate in this experiment, what we were able to see is that while the activation of RAC1 in this podocyte causes a collapse of the actin cytoskeleton, the podocytes that received this blocker of TRPC5, even though it wasn't a specific one, were actually able to kind of withstand the injury and maintain their integrity, which suggested that a TRPC5 inhibitor might be of relevance to maintaining the podocyte cytoskeleton and maintaining their intact phenotype. So again, I'm going to summarize the work that was done by these talented three young people in the lab, Konstantin, Thomas, and Suk Young. to basically say that we delineated a GPCR upstream of TRPC5 responsible for the activation of both RAC1 and ultimately TRPC5. RAC1 brings, through the vesicular insertion, TRPC5 channels into the plasma membrane. Due to the favorable electrochemical gradient, calcium comes into the cell, and that's sufficient in a localized way to activate RAC1 and keep this positive feedback loop going, which has detrimental effects for the podocyte. Well, this was intriguing, and we were able to do some work with the TRPC5 knockout mice in this JCI paper to demonstrate this. We actually wanted to take a step further. We wanted to know whether this pathway is actually relevant in an animal model that actually develops the full-blown human-like disease. In order to do that, and this is now brilliant work by two other very young and talented people in the lab, Yiming and Phil, we were able to develop a rat model. These are rats that have a podocyte-specific expression of the angiotensin type 1 receptor. So they're not in the vasculature, it's only in the podocytes. When you drive the expression of this AT1R, we were kind of agnostic. We didn't want to drive TRPC5, we wanted to drive the receptor and see what happens downstream to the channels, because there was this lingering question about TRPC6. So driving the AT1R definitely leads to a very human-like, progressive filter damage and kidney failure. These rats actually die of kidney failure late in life, as humans do as well. It takes a long time for this process to develop. So by the age of 50 weeks, these rats die of kidney failure. So we actually developed an approach for measuring single-channel activity in the glomeruli, which are the filtering units of the kidneys of these rats. So this is just a picture to demonstrate this. This is an isolated glomerulus from one of these rats. And this is the recording electrode landing on the podocytes, which are the cells sitting on the outside of this glomerulus. When you do experiments where the podocytes are labeled, one can easily see that these are the cells that are available for patch clamp at on the surface of this ball-like structure. We used the small molecule ribosol to activate TRPC5 channels, and we used the ML204 inhibitor that I showed you earlier to demonstrate whether we could have inhibition. If you take a wild-type rat under control conditions, whether they're early on in the disease onset, as we call it, these happen to be around 10-week-old rats, or whether you go into rats that have full-blown kidney disease, 20-week-old rats as shown here, the controls of those do not have a lot of TRPC5 activity. But when you go to the transgenic rats, what you can see is that the ribosol can invoke a TRPC5 what we think is a TRPC5 current based on the pharmacology here. ML204 is able to block this channel activity. And ribosol in the animals that have more advanced disease can actually lead to a very significant, if noisy, but significant activation of what we think is TRPC5 channels in these patches. We, of course, also did the control experiment looking for TRPC6. We used OAG for the activation of TRPC6. And as you can see, ML204, whether in control or AT1R transgenic rats, does not have an effect on the current. So suggesting that, we were basically looking at a progressive activation of TRPC5 channels in a way that could be summarized as such. I'm, again, pole vaulting through some additional experiments that are in the paper. But the basic crux of it was that we found a baseline, essentially phasic TRPC6 activity in the background that is there at all times. But with disease onset, we have a crescendo activity in TRPC5 channels, which also matches the development of the disease and the proteinuria, the faulty or leaky filters, as mentioned. This was actually reassuring to us that as we were doing this work, additional mutations were identified in TRPC6 where they were actually also now found to be loss of function mutations. So now imagine a scenario in which you have gain of function and loss of function mutations in the same channel causing the same disease. That would suggest that there is a homeostatic role for that channel, and that whether you have a lot more of its activity or none of its activity, you end up with disease. And indeed, this is the case. If we had more time, I would explain why. We have also studied that Rho-A is a homeostatic molecule that maintains the actin cytoskeleton in podocytes, and it depends on TRPC6 for its activity. And so that may explain this phenomenon. So with the knowledge that TRPC6 is homeostatic and that we really should be thinking about TRPC5 as part of this RAC-TRPC5 disease pathway, as I mentioned, we partnered with Corey Hopkins, a medicinal chemist, in an attempt to build a more specific TRPC5 inhibitor. I told you that ML204 actually blocks TRPC4 channels as well as TRPC5, and so we were keen to actually gain more specificity in our work. So this is how we came up with AC1903. Corey built several molecules, and we were able to eventually identify this. And when we looked, we actually found that indeed it seems to be much more specific for C5 versus C4 and C6, the two other channels that we were mostly worried about in the setting of this disease process that we were studying. Again, armed with this understanding now, we wanted to, of course, go in vivo, back into the rats, and demonstrate efficacy, hopefully. This is showing you here that a 1-week treatment with the drug is able to revert the phenotype back to baseline in these AT1R transgenic rats. And to be able to show target engagement, we actually repeated the experiments that I showed you earlier, this time using ribosol as the activator, but AC1903 as the blocker on the podocytes in the glomeruli of these rats as well, to demonstrate that we have adequate target engagement and blocking of the TRPC5 channels. Of course, I mentioned that the therapeutic hypothesis is to preserve podocytes, a podocyte preserving strategy. So of course, we had to go and show that as well. So this is a volumetric measurement looking at multiple histologic sections from kidneys from these rats to demonstrate that the rats that received the drug actually had a specific protection of their podocyte numbers, suggesting that this may be the first example of a true podocyte protective strategy. And armed with this further understanding, we were challenged appropriately by one of the reviewers to take a more severe disease model, the Dahl salt-sensitive rat, which has a specific known RAC1 activation as the driver of the disease, and to demonstrate there whether we could mitigate the phenotype. Just to give you an idea, these rats are here developing 5 to 10 times more severe and more rapid disease than the rats that I previously showed you. And even there, after 1 week's treatment in the setting of established disease, we are able to bend the curve downwards. So suggesting that, again, while we may not be able to replace every last podocyte, we're still able to ameliorate the disease in this case. What was also very significant is that in this model, this is not based on a lowering of the blood pressure, but rather hopefully a podocyte-specific effect. And this is demonstrated by the fact that the AC1903 did not actually affect the elevated blood pressure that is known to be there in this specific Dahl salt-sensitive rat model. So with this understanding, this is essentially a summary slide to say that we thought that TRPC5 inhibitors, there was now some evidence to suggest that they may be useful for suppressing progressive kidney disease of this flavor, the FSGS type, specifically dampening down this specific RAC TRPC5 disease pathway that I mentioned. There's a poster here to demonstrate some of the work that we have done since then, and Wan Chi, who's here in the audience, is actually one of my collaborators in the lab, together with Jen Pan, who's also in the audience. We work together at the Broad. And we're actually using the automated patch clamp, and I will not steal his thunder, it will be at his poster, but the idea is that we are also working now with Corey Hopkins to do some additional optimization, maintaining the specificity of the inhibitor that we found, but actually making it a more potent compound for TRPC5 with the goal to develop a better compound. And so Wan Chi can describe his work further using the automated patch clamp at the Broad. We, of course, were challenged. One could say, well, you've cured rats, but can you actually cure humans? And what is the evidence that this pathway is relevant to humans, or that the channel is relevant to human podocytes? And while, of course, only a clinical trial will ultimately demonstrate that, we actually were able to take pluripotent stem cells and build podocytes out of those based on established protocols. And Wan Chi, Choa is a graduate student in the lab who was responsible for making these iPSCs together with Wan Chi. And Wan Chi did some whole cell recordings, and Glarine is another together with ribosol, another very potent activator of TRPC5 channels. And what you can see here is that we have a very nice doubly rectifying current that is suggestive of a TRPC5 current in these now human podocytes for the first time. And you can see that AC1903 is able to block this current, so giving us a little bit of faith in the fact that we are likely to impact human podocytes as well as rat or mouse podocytes as we had done before. And indeed, it will not be long before we find out. So based on our work and the work of others who have confirmed a lot of our findings, the first person to be dosed with a TRPC5 inhibitor was just this past summer. And there's an ongoing clinical trial, which is funded, and I told you about my conflict, but funded by Goldfinch Bio, to demonstrate whether TRPC5 inhibitors will actually be useful in the clinic. And of course, I certainly hope on behalf of those many patients who suffer from this disease. So I would like to thank all the people in the lab who contribute greatly to making this work possible. Thank you for your attention, and I'll be glad to take any questions.
Thank you, Anna. Questions? You can see Bruce has his hand up here.
Bruce.
That's really nice, Anna.
Thank you.
I'm curious about TRPC5 in the brain and whether your inhibitors, do you design them to be peripherally restricted, or what do you think about the TRPC5?
That's a good question. So as demonstrated by the Clapham lab, the absence of TRPC5 channels results in an attenuated fear response in the mice. So the TRPC5 knockout mice are fearless. And so actually, as a result of that, it has been thought that TRPC5 inhibitors going into the brain might actually have efficacy as an anxiolytic or an antidepressant. And there have been some efforts in that regard, which are not in the clinic, as best I can tell, for reasons that I don't know. Now, that suggests to me that a TRPC5 inhibitor that would be targeted to the kidney, if it were to cross into the brain, which I think these are small molecules and they likely will, may have an on-target effect that may not be so adverse, if one can imagine. Of course, there's also this idea that these mice develop that phenotype because they are born without TRPC5 channels, and here we would basically be inhibiting, the channel in a child or a young adult. There are children with this disease, so one would hope that you could dose them as well. But hopefully you would not be actually inhibiting anything in the amygdala, which is the circuit that has been demonstrated to be defective in the knockout mice. So my feeling is that while these molecules are not peripherally restricted, that whatever amount of TRPC5 inhibitor gets into the brain may or may not actually have an adverse event. It seems less likely based on what we understand. The other note that I wanted to make is that, and this is in the PK study that we did for this paper, the amount of inhibitor that you need in order to target the podocyte is actually the exact opposite of what one needs to get into the brain. So the podocytes sit at the filter, so they see everything that is in the circulation. And as a result, delivering drugs to podocytes tends to be very easy, and you probably can get away with a much lower concentration. So there may be ways to actually not have a lot of crossing into the brain, because you can actually dose at a lower dose to affect the podocyte and not have an effect in the brain. So that would be another way to think about that. Of course, at the end, we have to see what happens in the clinic, but it seems hopeful. Oh, yes.
Yeah, sorry, the thing is, since you mentioned the effect of compounds in the brain, and you mentioned whether there are compounds that are in the clinic, I have to say, because I work on that program.
Oh.
So Hydra Biosciences actually had a program with Boehringer Ingelheim, and the goal of that program was actually the anxiety and fear-related diseases that you are mentioning, because that was the phenotype that was discovered when the TRPC5 knockout came out. And I have to say, that was the focus of the program, so we did not do kidney work with those compounds, but actually these compounds completely recapitulate what you see in the knockout. The compounds that were developed by Hydra and the collaboration with Boehringer Ingelheim are actually sub-nanomolar, and they are in the clinic.
Oh, brilliant.
Yeah.
Very good.
If you want to try it in your models, for example, there is a paper with one of them. It's a tool compound, but it's still like in the neighborhood of, it's under 1 nanomolar. It would be a very useful molecule to further test the validity of the TRPC5 as a target for kidney disease. But these things are in the clinic right now, phase 1.
Great. That's good.
No, it will be very interesting.
Yes, it will. Absolutely.
So I have a very quick question.
Sure.
So you mentioned that TRPC6 was important for homeostasis.
Mm-hmm.
Is the implication then that TRPC5 really does nothing useful?
Yeah, that's very interesting. Why did nature even put it there, right? It's a good question.
The RNA is expressed.
Yeah, so I think that while this is not something that we have done the definitive work to show, my explanation for the role of RAC, you could also ask, why do you need RAC, is that actually it is known to have a role in the formation of these interdigitating actin-rich structures that form the filter, and the understanding of the role of RAC in small phasic doses, so presumably TRPC5 and RAC would be needed in small phasic doses, would be to remodel that structure in the face of certain challenges, for example, an escalation in blood pressure in the setting of anger, or something like that, where physiologically our blood pressure changes, so that our filter doesn't break in the setting of that increased hydrostatic pressure that it would see. Now, that is a very hard measurement to actually make in real time, but there may be some advanced imaging approaches that could help with that. And so it would be worth testing for sure. It's an interesting idea.
Great, well, thank you.
Thanks so much.
Yep. We're going to move on. Sorry.